Hello, and welcome to episode 9 of the pharmaphorum podcast. My name's Dominic Tyre, and I'm pharmaphorum's creative and editorial director. Now for this episode, I met Alan Westwood from Matoke Pharma. They're a UK biotech that's looking to build on their work in re-engineered honey to develop novel pharmaceutical antimicrobials. Alan came to our offices in Surrey to talk about the research behind Matoke's Sergi Honey RO, an antimicrobial wound gel that has a CE mark in Europe, but also the next steps for its reactive oxygen component as a pharmaceutical product. With a background that includes 35 years at Bayer and stints in clinical research, sales and global marketing, he also gave his insights into the challenges of antibiotic research and the threat AMR poses. You can find more details on this episode, including a download link for the podcast and information about other instalments in the series at pharmaphorum.com/podcast. The podcast is also available on iTunes and now Spotify, Acast and Stitcher, where you can find and subscribe to it by searching for Pharmaphorum. Alan, welcome to the Pharmaphorum podcast. I'd like to begin with looking at your career today. You've had, of course, 35 or so years with Bayer, but your route into that wasn't necessarily a traditional pharmaceutical entry point, and then you've moved around quite a bit. So can you tell me a bit more about how you joined the industry?
Yes, thank you, Dominic. It has been a journey. I started it in Australia, studying as a microbiologist, and came to the UK back in the 70s, looking for a research career. Ultimately hoping to go back to Australia in some sort of academic capacity, but as things would happen, I drifted into work in the UK in research capacity. Worked for a famous British company called Tate & Lyle, fermenting things. In fact, ended up fermenting molasses and all sorts of sugars into alcohol, trying to develop an alternative to oil. Sort of worked. We had a nice process, but ultimately, Tate & Lyle, this was a bit too rich for them, and I was laid off, unfortunately. But in essence, it turned my career around, and I looked around for a while and actually joined Bayer as a sales representative, working in London for a couple of years. An incredible experience, which was useful in my latter life. And then they called me into head office to do clinical research. And I was part of the development team of ciprofloxacin, Cipro, part of the UK team that was part of the global team, and worked for 3 to 4 years on that. Basically submitted it in the UK. It was approved in the UK in 1987. And in the meantime, they'd sent me to Germany, to Leverkusen for Bayer Global, and I pretty much for the next 25 years was in the global part of Bayer, looking after, had little sidelines here and there, but effectively looking after anti-infectives for all that time.
So trained scientist, then into sales, back into research, from there into senior management roles. And you didn't stay in Leverkusen in all of that time? You moved around quite a bit.
Yeah, as I say, I went into global marketing. I was part of the launch team for Cipro, and that was in Germany, the UK, and the United States. And then they sent me to Latin America, where I was looking after the Central American marketing group, was launching ciprofloxacin and looking after other compounds. And then they called me back, had a little bit of a diversion, so I went to Abbott for a while in Chicago. Got fed up with Central America and all the challenges there.
Because you were there at a time of a fair bit of upheaval,
yes,
shall we say.
I was there during a war against the so-called terrorists then, who became the government after a while. Funny how that happens. But no, I left, I joined Abbott, and then after a year, Bayer called me back to Leverkusen, and I went back and effectively took over the global marketing, global strategic direction of the anti-infective portfolio Had a little stint in the United States, then launched Avalox, moxifloxacin, 1999. Moved me back to the United States as part of the global headquarters. Essentially, they're looking after Avalox, its launch. We then divested the US business to a company called Schering-Plough, which subsequently became Merck. So we lost Cipro and Avalox in the United States. So then the focus of Bayer was back ex-US in anti-infectives. Had to really fight very hard, I must say, to keep anti-infectives alive at Bayer, 26, 27, and changed it into not so much an anti-infective business, but tried to direct it more towards a pulmonology business.
And obviously you've had quite a ringside seat in terms of the pharmaceutical industry's approach to anti-infectives and antibiotics along the way. How have you seen companies' approach within this area change throughout that time? It's
been a heck of a challenge. Certainly, when you look, I think this is no secret, but anti-infectives, the business cases that are generated, and this has been for quite a while, if you like, since the primary care side of antibiotics has gone away. I mean, the whole primary care side of antibiotics is now effectively generic. The only thing is left for brands really is in the hospital serious infection area, and that, to be honest, is just not attractive, and it wasn't that attractive to Bayer. When you're faced with massive cardiovascular oncology assets, and you can see where the companies put their focus. So yes, I think, and this is where the debate really is firmly embedded now, is how we keep antibiotics alive, how we get big pharma involved again, and the recent the governmental 5-year plan on AMR is one initiative. There's lots of initiatives, don't get me wrong, but I think unless we do something soon, then big pharma will go further and further away. My experience has been essentially keeping antibiotics alive, going different directions. For example, one of the big challenges was, should we keep moving with quinolones after Cipro? A lot of quinolones had failed, and I think anybody in the field realizes a lot of toxicity issues, potential toxicity associated with quinolones. So facing that, I had to get the company in the first instance to continue with moxifloxacin, which they did, and it's been relatively successful globally. After that, we moved well out of quinolones and essentially into new chemical entities. I looked everywhere for new chemical entities, other opportunities. Either I didn't feel they were good enough or simply said other companies took them before me. But we then, at Bayer, went into inhalation antibiotics. We took our old ciprofloxacin, we tried to develop it for bronchiectasis. This was a dry powder technology, so essentially we're trying to prevent infections in the bronchiectasis patient. And then the other technology we licensed was a amikacin inhaled version, and this was associated with mechanical ventilation, so pneumonia in ventilated patients. And we had a vibrating mesh technology which would deliver high concentrations, we thought, of amikacin into the lung, to basically help the systemic antibiotics that were being used, for example, the cephalosporins, carbapenems, to give them a step up, as it were, in the very difficult pneumonia in that patient population. To cut to the end game, both of those programs unfortunately failed in phase 3. An enormous disappointment, because I personally felt that inhalation was the one. I still do, actually. I think that some of the study design, perhaps patient population, wasn't optimal. There's various reasons why these studies failed. But that's essentially what strategically I did. And to keep those assets alive and to keep them moving was a heck of a struggle in Bayer, to keep them, as it were, in management's eyes, attractive going forward. And the various ways I did that.
And it must have been really difficult when you've got both a high degree of risk in terms of the R&D side of it, and then a very challenging commercial landscape for these kind of products to be operating in at the moment. In terms of the R&D approach, how important do you think reformulation is to antibiotics, to be trying new approaches?
I think that one of the limits, where do you start? There's been various approaches here, but I think just basic new chemical entities, a lot of the time you are restricted by the dosing. We see it all the time in minor glycosides, perhaps something like tigecycline, one of the recent additions. If we could dose them higher, then perhaps we'd have a better chance, for example, against the development of resistance, not necessarily, and also giving us better clinical cures. So I think reformulation, people have tried liposomes and so forth, perhaps the nanotechnology or whatever, but inhalation is definitely one that I think that needs to be looked at. Hopefully, unlike Bayer, we can get some successes going forward.
I suppose that's one of the focuses for your current role now. So since towards the end of last year, 2018, you've been with Matoke Pharma,
exactly,
which is a British biotech company, a recently newly launched one. I think into company terms, it's not quite hit the 10-year mark, but it's pushing up against that. Can you tell me a bit about Matoke then?
Before I do that, I think one of my disappointments, obviously, is ending a career after having the highs of Cipro and the highs of Avalox, moxifloxacin, to have those phase 3 disappointments. It gave me even more energy and appetite to see if I could get something back on track. And therefore, looking at when I was approached by Matoke and their technology, yes, it did excite me, because the issues I was trying to handle at Bayer, or indeed what everybody's trying to handle, is this development of resistance, getting basically a higher standard of care into the various infection situations. I was still very much motivated to do that. So when Matoke came and started talking, yes, I looked at the technology. I based it also on the medical need that they were trying to look into. In this instance, the first instance, more in the chronic non-healing wound arena. That's where we'll apply what I'll talk about in a moment in the clinical setting. It looked very, very attractive, I must say.
And in terms of the company then, as we mentioned, it's a small to medium British biotech, but the team behind it, its genesis has quite an interesting story. It was set up by Ian Staples, who was a former managing director at Halfords.
Yes.
For listeners who haven't come across Halfords, it's a large UK retail chain. Ian's own background wasn't necessarily in retail, was it?
No, Ian was trained as a plant physiologist, but moved into that managerial commercial role for whatever reason. But he retired relatively early from that and went sailing into South America and plotted the coastline of the southern tip of South America.
Actually mapped.
Mapped a very complicated geography. But in doing so, he actually stumbled across, in Chile, a property which he then took over. And on that property, to cut a long story short, he actually started with beehives. In fact, I think he ended up with something like 600 beehives generating commercial honey. And again, this honey, as probably the people know, has been used in the treatment of wounds over many, many years, in fact thousands of years. He put some technology behind it, energized this honey, and in fact launched a product into the UK market called Surgery Honey, which is on the UK market as a device for the treatment of wounds. And that was basically Ian's background, and looking at how bees function, how bees protect their honey, and applying it to and developing a technology which is now coming. What I am doing with my team is turning this hopefully into a pharmaceutical product.
And within Matoke, you've got those working on Surgery Honey, and that on the UK market, it's registered as a medical device. And then, in addition to that, you've got the work that you're
the medical device is really being taken by several key clinicians in the UK, and they have used it in the clinical setting. And these are not control studies, but they're basically given what they think are spectacular results, certainly over and above what they use in their current standard of care. And it has given us the encouragement to take that and to turn this into a pharmaceutical entity and to essentially use the principles of what the medical device team has done, but to take out the variability and perhaps some of the inconsistencies, maybe in the honey or whatever, and put it into a true pharmaceutical development, right through to ultimately doing the randomized clinical studies.
And that product that you're working on, that's the reactive oxygen.
Yeah, the name is RO, is reactive oxygen 101, as we call it, and we're essentially taken the sugars and the enzyme and basically under certain conditions generating what we hope is an optimized concentration of the reactive oxygen, which is delivered in a topical gel to the site of the infection. I must say right from the get-go that this technology is not just going to be for chronic non-healing wounds. We believe, and again, when we get the appropriate resourcing, we can put it into other areas as well. For example, you could imagine rhinosinusitis, you can imagine chronic urinary tract infections, anywhere where there is this chronicity and the potential for multi-drug resistance organisms as well. Because this product is ultimately what we're trying to do here is to demonstrate through an optimized formulation is to deliver the reactive oxygen to kill bugs, including multi-drug resistant bugs, to penetrate biofilm, to hopefully regenerate the tissue, and to stimulate the immune system. So we are putting a lot of work into the formulation so that we can get what we believe is the optimal concentrations of the reactive oxygen to hit that target profile that we've defined for the formulation of killing the bugs, penetrating the biofilm, tissue regeneration immune, and basically then giving us the confidence to move into a preclinical program. Will be into the animals in the first instance to confirm what I've been talking about, and then into the sequence of proof of concept studies, and ultimately into random clinical trial.
And what's the timeline for that? What do you hope when you hope to hit those milestones?
Dominic, that's a good question. I think because we have this absolute background bedrock of open anecdotal with the Sergi honey, RO, but we believe we can accelerate. Because this is, again, it is topical, I have to be careful, but I think we can accelerate this, and I'd hope that we could get into into patients towards the end of this year.
And in terms of when it starts to be tested in patients, where are your greatest hopes for the product? Where do you think it's going to make the biggest difference?
Yeah. I think that's an excellent question. What we've been looking at very, very carefully is, if you like, what I call it, is that sweet spot of the type of patient you're looking for, ones that are, not too ill, or with this, particularly in the chronic non-healing wounds. There's some chronic non-healing wounds that will never heal, unfortunately, because of the patient's underlying comorbidities or whatever. And then, of course, there's the relatively easy ones that they'll get better without anything. So we've put a lot of effort with infectious disease clinicians, with wound experts, with surgeons into defining a patient population that we will in the first instance study, and then we'll align that with the regulatory authorities to make sure we got the right endpoints. For example, what I'm looking at here is how quickly we can heal the wound, so time to healing, whether it be complete healing or partial healing or whatever. But we must have always in the back of our mind, we must have a superiority approach where we can demonstrate that we're better than what's already available. Otherwise, why are we doing it?
Yeah. And how are those discussions with regulators and payers going? Obviously, there's a lot more focus nowadays, and perhaps over the last.
It's to be honest, it's in its infancy. We've had several We've had discussions. They're very encouraged. I think anybody who's looking at tackling this clinical indication, and B, obviously the offering of tackling antimicrobial resistance is very exciting to regulators. It's how we apply it then to a full pharmaceutical development is what we're working on over the next few months.
Mm-hmm. I suppose it must be heartening, though, to see, sitting here in the UK, seeing the leadership position, I suppose, that the UK is trying to take within antimicrobial resistance. That must help build your case.
Yeah, and we do a lot of work. We are doing a lot of work in the political arena. We are obviously trying to get monies from the various granting authorities. Not as easy as it sounds. I think that's the experience of a lot of SMEs. Obviously, we are trying to get the investment community excited about this opportunity. But yes, as a British biotech, absolutely, it's nice to have a leadership role. We also recognise that we want to use ultimately the various resources. We will, for example, some of the key wound experts are in the United States. I mean, it's a massive burden in the United States with just the underlying diabetic population there. So we are working with various universities in the United States, and of course we'll extend into Europe as well. So this is, yes, it's British originated, but the application, both in terms of the work we'll do and then ultimately hopefully the commercialisation will definitely be global. And you can imagine that it won't just stop at the United States and Europe, but you could see the potential here, the massive burden of wounds, but also of resistance in countries like China and of course Latin America.
And so you've obviously got some really big plans, big global plans. As a reasonably small British biotech company at the moment, with a fairly virtual structure to it as it stands, how are you going to achieve those plans? What does the roadmap look like for Matoke?
Yes, this is a question we ask ourselves every day. And it's got various opportunities or various ways we could go. Firstly, we need to generate resourcing to do all the studies I've been talking about. So we could envisage just pure investment money. We can talk about grants. We can talk about partnering with other small pharma. I don't think we're ready yet to go into the big pharma field, but I mean, ultimately, I could imagine that if we can generate the target profile that I've been talking about, then this is an extremely attractive opportunity for small to big pharma. In the meantime, however, as I say, we're looking to generate that profile that will excite them.
Mm-hmm. Well, it certainly sounds like it's a very exciting time to be working in this particular field.
It's absolutely, and everybody we talk to, I mean, we talk to a lot of the folk who are frustrated in this arena. And some of the market research we've been doing, just talking to infectious disease clinicians, surgeons, the people I mentioned, how frustrated they find themselves, and sometimes not even really knowing what the problems are, because it's not, as it were, as attractive as some of the other areas that they're working in. So I think if we can bring this entity in a little more top of mind, we'll get a lot more traction as well.
Okay. Well, Alan, thank you very much.
You're welcome.
And that brings to a close this episode of the pharmaphorum podcast and my chat with Matoke Pharma's Alan Westwood about his company's work in antimicrobial resistance. You can find more details of this episode, including a download link for the podcast and information about other instalments in the series at pharmaphorum.com/podcast. The podcast is also available on iTunes, as well as now Spotify, Acast and Stitcher, where you can find and subscribe to it by searching for pharmaphorum. And don't forget to visit our website to sign up for daily or weekly email, pharmaceutical news and analysis bulletins, and follow us on Twitter, where we are at pharmaphorum.